Hi, it's DrBiology here, and this video is on the discovery and development of drugs, and it's part of a series of videos on infection and response for AQA, GCSE biology and combined science, both higher tier and foundation tier. Okay, so first of all we need to think about, well, why do we need to test for drugs, and what are drugs? Well, drugs are medicines, or they are things that alter your body or your brain, and traditionally medicines have been extracted from plants. In particular, here's some examples. So digitalis, it's a heart drug. It originates from foxgloves. We've got aspirin, so that is a painkiller, and that originates from the willow tree. And penicillin, that we talked about in a previous video, originates from the mold Penicillium. So we've got some foxgloves, willow tree, and a bit of Penicillium notatum. So developing new drugs. Why do we have to develop new drugs? Well, we need to test them intensively for these three following reasons. Okay, so we need to check its toxicity. So that basically means, is it harmful? We need to test efficacy. So does it work? The dose, what amount is safe and effective to give. There are some really important keywords in this section of work as well that will come up time and again. So let's just have a look at them. So first of all is placebo. So a placebo is a tablet that does not contain the drug or new medicine. So often the placebo will contain a different drug that is already used to treat the disease. And something called a double-blind trial. So a double-blind trial is where the patients are either given a placebo or the drug, but most importantly, neither the doctor nor the patient knows who has received the real drug or placebo until the end of the trial. Sometimes they don't even tell you at the end of the trial, and that's to reduce bias, and we'll talk about bias in a minute. So all drugs that are for sale in, for example, chemists, or are being prescribed by doctors or hospitals, they have to go through some trials. First of all, really to see whether they work, but also to look at their safety and their toxicity. So these are the main stages. The stages slightly change from different examples, but these are the main stages. So first of all, you get preclinical trials. So these are when drugs are tested on cells, tissues, and eventually animals. So they're testing lots and lots of different types of drug to see whether they actually are safe, but also whether they work. Then you get clinical trials, so you start to use it on humans, and the first set of humans are healthy volunteers. So what you do is you give them low doses, and you're really testing the safety of that drug, not testing whether it's actually working, but testing whether it's safe to use. Secondly, if it's safe, then a large number of patients are used to determine the optimum dose. So they'll give it to patients that volunteer, and they will then work out what's the correct dose without producing too many side effects. And then they will eventually do what we call double-blind trials. So they're used where neither the patient nor the doctor know who has had the real drug or the placebo. And the reason they're doing that is to avoid bias. So they're really trying to see, is it the drug that is making people get better? And then thirdly, it will then go to what we call peer review. So all that data, all that information needs to go to independent bodies, government bodies, international bodies, and some of it will be published as well, and the results are checked by independent professional researchers. There is a fourth step, and that is when the drug has been actually released, then further studies will continue to go on for quite some time to see if there's any longer term side effects. So as I said, results are usually published in what we call peer-reviewed journals. So other scientists check on these results. And then you've got bodies such as the National Institute for Clinical Excellence. They look at the published results and they decide which drugs give good value for money and can be prescribed by people like the NHS. So obviously I've gone very quickly through these stages, so what I'd like you to do is to see if you can put these statements in order. So what I suggest you do is you pause it, read it and answer it. So in 3, 2, 1. Okay, so let's look at the answers. So firstly, drugs are tested on human cells and tissues. This can't be done if the drug affects the whole or multiple body systems. Secondly, the next step is to test the drug on live animals. This is to see whether the drug works, find out about its toxicity and work out the dosage. If it passes that, it's then tested on human volunteers, and these are healthy volunteers in a clinical trial. And this is to make sure that it does not have any harmful effects when the body is working normally. So a low dosage is usually given and it then is gradually increased. Then they're tested on people suffering from the illness itself. So they need to try and find the optimum dose. So that's the dose that's most effective with few side effects. Then patients are put into two groups. One is given the new drug and the other is given a placebo, so a substance that's like the drug being tested but it isn't. So this is so the doctor can see the difference the drug actually makes. Clinical trials are blind as well, so the patient doesn't know if they have been given the drug or the placebo. So it's a double-blind trial if both the patient and the doctor don't know who's had the treatment. And then, as it says there, so neither the patient nor the doctor knows which group have been given the drug. This stops either being influenced by their knowledge, and it does give the most accurate results. Okay, so let's go through a question checkpoint. If you haven't seen these before, what I ask you is to pause, read and answer, and then I'll go through the answers. So pause in 3, 2, 1. Right, okay, question 1. So list A gives the name of 3 stages in the trialing of a new drug. List B gives information about the 3 stages. Draw a line from each stage in list A to the correct information in list B. So you need to look at list A and work out which is the correct one in list B. So testing on humans includes a placebo. It's used to prove that the drug is effective on humans. Tests on humans using very small quantities of the drug, well, that's the first stage in the clinical trials of the drug. And then tests on animals is used to see if the drug is toxic. Question 1B. So again, I'd like you to pause, read and answer in 3, 2, 1. Okay, so they talk about 80 volunteers being, they're asked to report any side effects. And they show the results, so you can see the reported effects. And it says, based on the results of this trial, what should the drug company do next? Well, they should stop the trial. The reason being is that there are lots of side effects, okay? Give a reason. The side effects are too severe. So you're actually getting a large proportion, that over half, who are severely sick, which is probably not a good thing for a new drug. Question 2. Okay, this is a bit more involved. So again, I'd like you to pause it, read it very carefully and answer. So in 3, 2, 1. Okay, so they give you a graph, and graph shows how much time the different stages of testing took for a new drug. And they do tell you which are the clinical trials. And it says how much more time did the clinical trials take than the laboratory testing. So what you got to do is add it all up and minus the laboratory testing. So you would have got 5 years. Apart from the time taken, what are the differences there between laboratory testing and clinical trials? Well, the lab tests are on cells, tissues or animals, or you could say the clinical trials are using humans, not humans, humans. Okay, B, so again, and I'd like you to pause, read and answer in 3, 2, 1. Okay. So during phase 1 clinical trials, the drug is tested on healthy volunteers using low doses. Suggest why only healthy volunteers and only low doses are used at this stage of drug testing. So there are 2 marks here. So they're looking any one for the first mark. that it's too great a risk. If you're ill or you're the patient, you don't want to get worse. The patients could be taking other drugs which would affect your results. And also, if you're using a healthy volunteer, if there are side effects, you're more likely to see them in that volunteer than the patient. And then second mark, reason why it's a low dose is to reduce the risk. Okay, and also to look for side effects or reduce side effects as well. So in phase 2 and phase 3 clinical trials, a double-blind trial is usually done. Explain what a double-blind trial is and why it's a good practice. Well, 3 marks. So you've got to say, for the first mark, you've got to mention that a placebo and the drug are both tested. The key thing is neither the patient nor the doctor knows. Well, why is that good? Well, it avoids bias. You could have it controls a psychological effect. That's quite interesting because sometimes people say they feel better just because they've been told or they've been given something that they think will make them better, but it doesn't. It's just a psychological effect. And also you're telling if it's the drug that is working rather than the placebo effect. Okay, question 3. So again, I'd like you to pause, read and answer in 3, 2, 1. Okay, so in this they show you a diagram, a timeline for the testing of the new drugs, and you can see how long it takes usually. Obviously the more recent COVID vaccine was much quicker within the year because there are many different companies doing the work simultaneously. So whilst they were doing preclinical testing, they were also doing clinical testing as well. Well, after they checked that it was safe. So it says, what is the main purpose of preclinical testing? So yeah, testing for toxicity, seeing if it's safe, see if it works. B, what is the main purpose of phase 1? Well, phase 1 is to test for any side effects. In phase 1 testing, healthy volunteers are used. Suggest one reason. Well, this is again 1 mark, so it could be higher risk for patients. Patients could be on other drugs. It allows you to see the side effects more clearly. So you can see we're getting the same kind of answers coming through in the different questions. Okay, so again, I'd like you to pause, read and answer in 3, 2, 1. So what is the main purpose of the phase 2 and phase 3 testing? Well, again, you want to find an optimum dose, or you could say the results are more reliable, or you're checking to see whether the drug actually works. What is meant by placebo? It's a tablet or drug that does not contain the drug. It looks exactly the same though. And then it says, during the testing, who knows which patients are receiving the placebo? Okay, well, neither the patients nor doctors because it's a double-blind trial. Okay, so this brings us to the end of this video. I hope you found it useful. Please do like it, please do subscribe, and please do put your bell alert reminders for more videos coming soon. I'll see you soon.